the ID. That's the problem I had with Wolfgang's talk. I only put the proper password in, but it wanted me to put everything in on you. I don't know why.
So I think we're okay.
The ID.
All right, so it looks like it's working. Um, all right, so I wanted to say hello, everyone. Thank you for being here for another fine seminar series. My name is Lauren Hayes. I'm a professor at the University of Tennessee at Chattanooga, and I'm your host today. Quick a couple announcements before we get started. First off, we wanted to say this is the 8th season of the Fine, and we've crossed over 100 seminars since we began this back at the early days of the pandemic. And personally, I want to thank all of you because I honestly, it wouldn't be a success without people's participation. I think it's been great to have so many people joining us regularly, and I'm glad to see everyone here again. Um, I think the organizers, Karsten, Eduardo, and Adriana would agree that this has been a really fruitful experience for all of us. And we're really happy to have everyone here. We also want to thank a couple of people behind the scenes. Um, Logan Platt, my graduate student, has been working on cleaning up the YouTube page and organizing it. So if you want to go back and watch Fine seminars or have your students watch it, he's working on organizing that so it's a little more user friendly. And particularly, I want to thank Facundo Fernandez Duque. Facundo developed the website that we use for this Fine, and it looks remarkable. And he continuously maintains it for us, and we're thankful for his help on that process. And again, it really makes the organizers' lives a little bit easier as we try to navigate this. Regarding the website, I want to point it out to you for those of you who are new. So I'm going to make an attempt to share my screen here. Of course, it's gone. What would the first find be without some technical issues? Let's see if we can get it to work this time. There we go. So hopefully you can see the screen. I've got the Fine website up here. For those of you new, those of you who've been around for a while, maybe you can use some of the resources. We have all the past speakers and links to their YouTube links to their talks, also papers that they provided. As you know, every speaker gives us 3 PDFs that you can read ahead of time. For those of you interested in using resources for teaching, there's a link here, Find Teaching. In there, we have some lecture slides. We also have a link to a paper that we published on how to use remote seminars and teaching. And there's other resources in there for you if you want to integrate this into your classrooms. In terms of the web, we also have a link to the current speakers, which brings me to where we're going. If you click on this link here, you can see that we have a really great seminar series lined up for this season. And it's going to start with Connor Philson, which I'll talk about in just a moment. But you can see that next week we have Elise Houchard talking about power asymmetries in primates. And I'll just quickly scroll down so that you get a sense of what's coming this series. Lots of interesting talks coming from, we try to bring in speakers that are prominent, but also we try to bring in some of the rising stars, people in the field who are getting their feet, getting their standing in the field and are doing some exciting stuff. We hope to promote their work so that they can progress successfully in their careers. And you can see lots of different themes coming through this series, lots of different people from different countries, and we're looking forward to having this this Fine coming up. And so that brings me to today's speaker through here. So today I'm really excited to introduce to you Connor Philson. Connor is a postdoc at the University of Exeter in England. He earned his PhD at the University of California, Los Angeles, just in 2023. So he's a rising star in the field, and we're glad to have him with us today. His current postdoc, as I mentioned, is at Exeter. He's working with Laurent Brent and at the Center for Research in Animal Behavior. He's interested in a number of things, including the drivers and consequences and evolution of social behavior. And he focuses on things like the individual social position and group social structure, taking a multi-level selection perspective. If you look at his CV, he has a pretty impressive publication record at this early career stage, about 15 papers in journals like Current Zoology, Royal Open Society, Animal Behavior, Biology Letters, Behavioral Ecology. And notably, he's mentored quite a number of students in his research, including 18 since 2021, and many of them have helped to publish. And then he's also active in outreach issues, in particularly addressing science policy and use of field stations. He's an assistant editor-in-chief for the Journal of Science, Policy, and Governance, and is the board of directors for the Organization of Biological Field Stations. So quite a broad spectrum of things that he's contributing to in our field. And today, Connor's going to be speaking to us about his research. The title of this talk is Evolution of Social Position and Structure, Multi-Level Sex Selection Pressure. And with that, I'm going to stop sharing the screen and welcome Connor to the seminar series. And thanks for being here with us today.
Well, thank you very much for having me. This is very exciting to kick off another session. Hopefully you are able to see my screen at this point. Fantastic. Well, yeah, thank you very much for having me. I remember watching these back in 2022 or in 2020 during the pandemic, which isn't that long ago, feels very long ago. So it's quite an honor to be here to help kick off what you said, the eighth series of it. So I appreciate you guys tuning in. So the evolution of social organization, the evolution of animal societies is an important question because social organization has ranging implications from influencing individual fitness outcomes and community health to how individuals support others and tackle shared challenges like resource acquisition or avoiding predators, and up to implications about how we govern ourselves and structure our world. And societies are really complex, consisting of many levels of social organization nested within each other, such as individuals working in research groups and research groups housed in departments, and these departments comprising the larger university. And so when thinking about the evolution of societies, two of the most important fundamental levels of social organization are that of the individual socially interacting with others, and that of the group social structure that emerges from these repeated dyadic interactions between individuals within the group. Not all individuals are as social as others, and not all groups are the same. An individual's social position or the individual's social phenotype depends on their own behavior, but also that of others, whereas the group's social structure is an emergent property, emergent phenotype of all the individual level interactions. And so within social groups, individuals vary in their pattern of social interactions with others, and this individual variation in part drives differences in the emergent group social structure. And so we can ask not only about how social individuals are, about their position within the group, and how this impacts their fitness, but we can also ask about how social groups are structured and how this structure impacts each individual who resides in that group. And so to understand the evolution of societies and social organization, we must understand if both of these two levels of social organization are subject to evolution by natural selection. And to do so, both individual social position and group social structure must meet the triad of requirements for evolution by natural selection to occur. That is, at both levels, there must be variation of a trait, replication of this trait across generations, and differential fitness outcomes of this trait, or selection for this trait. And this triad has been well explored on the individual level in a wide range of species, with most species experiencing positive selection for individual social connectivity, more central positions, gaining individual health and survival benefits. But the triad has not been as well studied for both the individual and the group levels within the same species. And thus we're missing a part of the story when exploring the evolution of social organization and the evolution of society. But of course, these two levels of social organization are fundamentally nested, like most biological traits are, social or not. In nested structures, the levels may feed back to influence each other linearly or non-linearly. For sociality, individuals may change who they socially interact with or how frequently, which may restructure the whole group. At the group level, changes in social structure may disproportionately harm or benefit some individuals within the group, resulting in changes in the distribution of individual social behaviors. Said simply, individuals structure social groups, and these social groups feed back to influence patterns of individual social interactions. And so because these individual and group social phenotypes are nested, natural selection at one level may very likely affect the other level, either directly or indirectly. And thus, when we're exploring either of these two levels independently, we run the risk of potentially falsely detecting selection at the level we're measuring, when actually some of that selection might be on the other level, but is being detected at the level we're measuring. This means we could be detecting some group level selection at the individual level, or vice versa, some individual level selection at the group level. Or we could be experiencing type 2 errors, where we're not detecting any selection at the level we're measuring, but selection is occurring at the level we're not currently measuring. So because these two levels of social organization are nested, we have to ask ourselves how much of the selection at the individual level that we found in all of these species is actually selection at the group level that is just being detected at the individual level. So in order to more comprehensively understand the evolution of social organization in animal societies, after the triad of requirements for evolution have been met at each of the levels, selection can then be quantified not at the individual level or the group level independently, but the two levels can be quantified simultaneously in order to partition natural selection among the levels relative to each other. Multi-level selection does just this. Multi-level selection is a theoretical framework positing that natural selection occurs at multiple levels of biological organization at once, and at levels other than only the gene. Multi-level selection is a framework for understanding the vast selective forces acting on phenotypes and individuals, and does not pit individual level selection against group level selection, but instead proposes selection can simultaneously occur and be measured at multiple levels at once. This could be selection acting simultaneously at the level of the gene in cells or groups of cells, like organs or cancer, or at the level of the individual organism in the group, from microbiomes to social groups. Multi-level selection has been documented as a driving force in the emergence of the first multicellular organisms to the structure of entire ecosystems in human cultures. Multi-level selection also acts on morphological phenotypes in plants and in animals. So any level of biological organization that meets the triad of variation, replication, and individual fitness consequences is subject to the evolution by natural selection. But for nested levels of organization, like sociality, multi-level selection may be a more comprehensive and accurate way to understand natural selection, as it accounts for the multiple forces of natural selection and accounts for these multiple levels relative to each other. And the evolutionary consequences of multi-level selection are potentially striking. Higher level selection in the same direction as individual level selection could increase the rate of the evolutionary response, but higher level selection in the opposite direction could reduce, flatten, or even reverse the evolutionary response to selection. And so with multi-level selection, we can develop a more comprehensive view of the evolution of nested structures. And I want to make this very clear. This is very important. At both levels of social organization, I'm talking about the fitness consequences for the individual. I'm interested in how social individuals are because that affects them. And I'm interested in how social the group is because that affects each individual within the group. I'm talking about multi-level selection, which posits any phenotypic level that is driving fitness variation at the lowest level being explored, in this case, the individual, and that may be under natural selection. I am not talking about groups competing with groups for success or proliferation. I'm not talking about group selection, perhaps in its more traditional sense. For those of you familiar with multi-level selection, this is more in line with the MLS1 framework. This is not really the MLS2 framework. And so with multi-level selection, we can partition selection relatively among nested levels of social organization to more comprehensively quantify social evolution. However, despite social behavior being a very common theoretical example in debates about multi-level selection, no work has really explored the presence and strength of multi-level selection for both individual social behavior and group social structure in wild and free-living animals. Some really inspiring work in laboratory and experimental settings has explored multi-level sexual conflict in water striders and for social behaviors with forked fungus beetles. But in the absence of evidence for multi-level selection for social behavior in the wild, this decades-old debate of multi-level selection remains largely a theoretical and not empirical debate. And we can use social network analysis to precisely quantify both of these levels of social organization. Networks are a simple way to represent complex behaviors where we have lines representing social interactions between individuals. And with social network analysis, we can ask not just how many social partners this individual has, but how frequent are those social interactions? Which of their connections are connected to each other? And about their position in their larger social group, incorporating both their direct and indirect partners. And for the group's social structure, we can ask how reciprocal the group is, or how polarized, how breakable, how homogeneous, or how connected or sparsely connected the group is. And so with social network analysis, we're able to precisely quantify the two levels of social organization to ask about multi-level selection. So if there is to be evolution of social position and social structure, we must first complete the triad of requirements, variation, replication, and selection, and this must be completed at both levels. Then, if we have met these three requirements at both levels, then evolution by natural selection for both levels is possible. But because social position and structure, like most nested biological traits, are not completely independent levels, we should not only explore these levels independently, we should also explore selection for social position and social structure concurrently and relative to each other. So if we modify our requirements for evolution just a little, we're looking for variation, replication, selection, and then multi-level selection. So today I'm going to tell you a story in 4 parts. First, we'll explore the variation of social position and structure and explore some of the factors that drive this variation. Second, we'll ask if social position and social structure are heritable. Third, we'll quantify the individual fitness consequences of the two levels independently. And then lastly, we'll put it all together to estimate the relative strength of multi-level selection for social position and social structure. And we're going to explore these 4 parts in the yellow-belly marmot. A lot is already known about this species on the individual level, and I will walk you through what we do know. But in short, we know these marmots are weird. Whereas other species on this list on your screen experience mostly benefits from increased individual social connectivity, marmots experience mostly costs. And so perhaps in this species that experience mostly costs on the individual level, maybe there's more positive selection for sociality at the group level in this system. And thus it's really a great species to ask these questions in. So this is the yellow-belly marmot, Marmota flavirventor. Hereby, I'll just call them marmots. They're harem polygynous, where one male tries to control multiple matrilines from whom the social groups are formed around. They're obligate winter hibernators, and they're facultatively social, meaning they're found in a wide range of group sizes and sometimes alone. And some 96% of their summer mortality is attributable to predation, which is one of the possible explanations for why these marmots live in social groups. So the marmot annual cycle is essentially wake up in spring, reproduce and get fat over the summer, and then begin hibernation again in the fall. And yearlings disperse around midsummer, right before the pups come up. And so yearlings and adults have slightly different life history strategies. And because body mass is vital for a successful hibernation, essentially it's a game of get fat or die. And get fat or die is so important in this system that after I spent a day trapping and watching marmots with a Nat Geo journalist, talking about shifting phenologies and mismatches in marmot hibernation, talking about climate and snow patterns in the Rockies, and the value of long-term field studies and field stations to understand biology and conservation. This is the first quote that they chose So even though all that other stuff is really important, get fat or die remains, get fat or die is still important. So I study the population at and around the Rocky Mountain Biological Laboratory, which is an incredible biological field station in Western Colorado. It's up around 9,500 feet or 2,900 meters above sea level, where we've been studying them annually since 1962. So in the last 60 plus years, we have over 4,500 unique individuals, over 200,000 behavioral observations, and just since 2002, over 23,000 hours of observation. And these marmots live in colonies in two primary areas, at lower elevation and at higher elevation sites. Those higher elevation sites experience more harsh weather conditions comparatively, and winter lasts about 2 weeks longer at those higher elevation sites than at those lower elevation sites. And this likely makes the Rumble Marmot Project the second longest running project on wild and free living mammals in the world. So it's quite the powerful data set. And to collect the data on these animals for 5 months a year, we go out 6 days a week, often twice a day, to observe and trap these animals. During this, we uniquely mark all of the individuals, take a suite of data for physiological, hormonal, health, and genetic analyses, and we record a suite of behavioral data. So we know who's where, who's interacting with who's initiating and who's receiving those interactions, what predators are around, and the daily environmental conditions. And these marmots exhibit a wide variety of social behaviors, from sitting together, looking for predators, to greeting each other, to allo grooming, and to foraging together, to playful biting, and to all-out tackling each other. And so with this variety of behaviors, I construct their social networks. The networks I'll talk about today are all affiliative interactions, affiliative networks, which represent some 88% of all of their social behaviors. We can also build agonistic social networks. Those are more sparse. We use those to ask different questions, and I won't focus on agonistic interactions today. And the affiliative networks I build include yearlings and adults and are based on space use overlap. And this project is really the emergent product of hundreds of individuals across the past 61, 62 years, especially Kent Armitage, who started the project back in 1962, Julian Martin, who co-PIs the project currently, and Dan Blumstein, the current project PI. And some of the work I'm presenting today comes from this incredible group of people, including 5 rockstar undergraduates who I supervised and whose work I will feature today. And if you're a longtime FIND participant, you'll remember back in fall 2020 and spring 2021, the first 2 seasons of the FIND seminar, Dan Blumstein and Adriana both gave incredible talks about these marmots. So now 3 years on, I think it's time for a marmot update, but with a multi-level selection perspective. So that's the system. Lots of data, uniquely marked individuals, known genealogies. We know a good amount about their biology. They have interesting seasonal patterns and a variety of social behaviors. Now, is there variation in social position and social structure? And if so, what are some of the factors that drive this variation? Marmot social groups, even in the same place, vary from year to year. And so these are small examples of marmot social networks, social groups, and they can get as large as 30 individuals, but that is less common and much harder for you to see the differences in. So these are just small examples. But as you can see here, individuals may be more or less connected or isolatable than other individuals, and social groups vary in their pattern and structure of connectivity. And because this is a matrilineal society, these groups are largely kin-structured, and because dispersal often results in individuals moving out of the valley and out of our study area, the between-group relatedness is quite low. So, yes, there is variation in social position and social structure. But what are some of the factors driving this variation? Individual social position, group social structure are both social phenotypes, and thus with our classic equation, we're specifically interested in the genetic and the environmental drivers, because these 2 factors underlie the social phenotype from which selection can occur. So first, let's explore environmental drivers, both abiotic and then biotic. Marmots are a seasonal hibernator living in the high alpine, where the summer and the winters are quite starkly different. And so to quantify the abiotic environment, we can differentiate between weather, like precipitation, temperature, and humidity, from the timing and length of the two seasons. So to ask this question, I supervised an undergraduate student, Carla. We asked about the relationship between measures of individual social position and both weather and seasonal timing. And from this, 3 core relationships stood out that I want to share with you today. All 3 of these relationships are with clustering coefficient, which measures the extent an individual is socially clustered together with others, or the individual social clickiness of an individual. And so as we move up our y-axis, we have more social connectivity, more clickiness. An individual clustering coefficient is important for the structure of social groups. So the results I'm about to share may matter across our 2 levels of social organization, though we didn't explore the group directly in this project. And clustering coefficient was associated with 3 different measures of seasonality. The length of the growing season before hibernation, which is a proxy for how much time the marmots had to eat and get fat before hibernation. The length of winter, which is a proxy for the length of hibernation, with longer winters requiring more body mass. And the date of 50% snowmelt, which is a proxy for when marmots emerge from hibernation, as this is the end of winter and the start of the growing season when there is exposed vegetation for the marmots to eat. We did not find any relationship with weather, only seasonal length and seasonal timing. So we found a positive relationship between social connectivity and growing season length. That is, when there was more time to eat and get fat in the previous summer, marmots were more socially clicky the following spring. This maybe is because they emerged from hibernation with more body mass. We also found a positive relationship between social connectivity and winter's length. That is, marmots were more clicky in a spring after a long winter hibernation. And maybe this is because the time frame to start interacting and reproduce is more compressed after a longer winter. But we found a negative relationship between social connectivity and the timing of winter's end and spring's beginning. That is, the longer there is snow on the ground in spring, the less social and clicky individuals are. Maybe this is because when there's snow on the ground and the marmots aren't outside their burrow eating and interacting as much, they're spending more time in their burrows where we can't actually see what they're doing. So given climate change is making abiotic environmental conditions more stochastic and driving mismatches in seasonal timing and phenology, especially in high elevation populations like these marmots, this is an important relationship to understand. There is much more work to be done, both in this system and across taxa. So that was the abiotic environment. What about the biotic environmental drivers? The diversity and composition of the gut microbiome in its relation to social behavior has been well studied in laboratory rodent systems, and thus the gut microbiome could be an important link in explaining some variation in individual social behavior in the wild. So supervising another rockstar undergraduate, Madison, we used a few different measures of individual social position to explore sociality's relationship with a dozen gut microbes with known physiological, immunological, and behavioral associations in these laboratory rodent systems. So here are just 2 of the results. On our y-axis on the left, there is, again, clustering coefficient, how clicky an individual is. And on the other y-axis is embeddedness, which quantifies how central and connected an individual is within their whole group. And on our x-axis, we have Streptococcus, which is a genus of bacteria related to strep throat in humans. And we found that greater abundance of this strep throat-related gut bacteria is associated with decreased social connectivity and position. And perhaps we can interpret this as more sick marmots, or marmots that were once sick and now have higher abundances of this bacteria in their gut, potentially being less central and less connected in their groups. And if you've ever had strep throat like I have, I think you can understand that perhaps you would be less social when you were sick or if you were sick. And so with these two projects, we know some of the environmental drivers of social variation seen in this system. And so we can check off the first requirement of our triad. There is indeed variation. Now, is there a genetic component to the social phenotype in this system? Lay et al. In 2010 used a quantitative genetics approach, an animal model, to estimate the additive genetic variance of social position. In other words, is individual social position measured with social network analysis heritable? And the answer is yes. Genetics alone explains 10 to 20% of variation in some social traits. Another way of saying this is that marmots have a genetic predisposition to their social behavior. Social position is indeed heritable across generations. So this genetic component plus the environmental component gives us an insight into how the social phenotype is generated. But the drivers of the social phenotype I've just shown you, both genetic and environmental, were for individual social position. What about the drivers of the group social phenotype? Let's remember, individuals structure groups, and these social groups feed back to influence patterns of individual social interactions. Group social structure is an emergent phenotype of all the individual level interactions. And thus, group social structure is a product of all the environmental pressures individuals experience and is a product of all the individual's genotypes. So because social position and structure are nested, group social structures are logically heritable via the genetics of the individuals who comprise the group, but much more work on this is needed. We need to better understand the genetic components of group social structure directly. And we need to further tease apart how the environment, both biotic and abiotic, can act directly on social group structure. Some really neat work in other non-marmot systems have already begun exploring how the environment and population relate to social group structure. So more of that type of work is needed to understand group social structure more directly. So we have variation at both levels of social organization. We have replication via genetic heritability at both levels, but we're acknowledging that more work is needed to specifically understand group level genetic heritability. So we're building the case towards evolution. So let's move on to explore the consequences of this variation at two levels, at both of the levels of social organization. First, each level independently, and then with our multi-level selection perspective. So first, exploring the individual level fitness consequences of both levels of social organization independently will give us an insight into these how the more comprehensive multi-level selection might differ. So since 2009, the fitness consequences of individual social position have been well explored in a variety of ways in the system, all with social network analysis, to illuminate the costs and the benefits of individual social positions. The result I'm about to show you are the fitness consequences of increased individual sociality. And these were measured with a variety of social network measures, but for simplicity's sake, I'm just going to cover the consequences. So we know that more social females have fewer kids. More social marmots, both males and females, have decreased hibernation survival. We know more social marmots, both males and females, don't live as long. They have shorter lifespans. So what about some of the benefits? More social females have increased summer survival, and more social yearlings are less likely to disperse away from their natal colony. So there are costs for reproductive success, hibernation survival, and lifespan, but there are benefits for summer survival and philopatry. And the dispersal finding tells us that social position is not only influencing fitness consequences, but also major life history decisions too. So in short, more social connectivity has some costs and benefits, but generally social connectivity is costly. To build on this story of the consequences for individuals just a little more, I co-supervised an undergraduate tally in the field out in Colorado this past summer. We were interested in another individual decision, when to flee from an approaching threat. The social security hypothesis predicts that more socially connected individuals would perceive less fear and thus flee from an approaching threat at a shorter distance. This is especially relevant because marmots are quite the tasty prey item, and marmot social group living may be contributable and may be driven by anti-predator benefits. So how connected you are within your group could matter. And it does. Individuals with more social interactions perceive less risk. And this is a plot for the marginal effects. And this, I will acknowledge, is a very modest relationship, but it is significant even when controlling for other biologically relevant variables in the model. And so maybe you're thinking, hopefully you're thinking, that seems to be a lot of costs to increased social connectedness, and it was. Remember, marmots are kind of weird. They're relatively unique in the consequences of individual social position. Most species experience largely benefits to increase connectivity in their social group. Marmots are kind of the outlier. Perhaps this is attributable to their facultatively social nature, as the other species on this list are mostly obligates in terms of their social behavior. They're more gregarious than these marmots. And so we know there are individual fitness consequences of an individual's social behavior and position in their network, and that being more social and more connected is mostly costly. So if the individual level is mostly costly, maybe there are benefits of the group social structure for the individuals who reside in those groups. So at the group level, first looking at reproductive success, on our y-axis is the number of offspring adult females weaned in a given year. On our x-axis is cut points, which tells us how many social relationships in the network have to be broken or lost in order to result in the group fragmenting into 2 or more social groups. So as we move down the x-axis, more social relationships have to be broken, and thus the group is more strongly connected as we move down our x-axis. I found that females who live in more socially connected groups have fewer kids. That is, females residing in groups that are less easily fragmented into 2 or more social groups wean fewer kids. This is, again, a very modest association. It shows that some attributes of the group influence individual fitness even after controlling for other biologically relevant variables like body condition, location, age, individual ID, and year. Now, what about summer survival? Predation, again, counts for about 96% of summer mortality in this population, and given predator avoidance is a likely driver, or one of the many likely drivers of marmot social group living, we could predict that individuals residing in more socially connected groups would be more likely to survive summer because these well-connected groups may be better at detecting predators and responding to alarm calls. But we found no relationship for summer survival in any of our measures of the group social phenotype. And perhaps this is surprising given the positive survival benefits of increased social connectivity we found at the individual level. So, social structure does not relate to summer survival, but what about winter hibernation survival? Given marmots spend the summer balancing avoiding predators and gaining body mass for hibernation and socially interacting with each other, they have quite the time budget to balance. So we could predict that individuals residing in more connected and social groups would have less time to gain mass and thus would have decreased hibernation survival. Alternatively, we could predict that more connected social groups are more likely to socially hibernate, boosting thermoregulatory benefits and thus increasing hibernation survival. So looking at winter survival, we again have our cut points measure, which as we move down the x-axis represents how many social relationships must be broken in order to have the group fragment into 2 or more separate social groups. And the groups are more connected as we move down our x-axis. I found that individuals residing in more connected groups have higher hibernation survival. So this supports our prediction about potential social hibernation, as groups that are more breakable may not socially hibernate with as many individuals. So as we move down our x-axis, we have these groups that are harder to break and thus might be hibernating socially. This result is also surprising for 2 reasons. One, because the relationship for individual level of sociality and hibernation was in the opposite direction. And two, we also just observed the opposite relationship for reproductive success and cut points. Again, a modest relationship, but statistically significant, suggests that group social structure matters in different ways depending on the fitness context. So, with all of this, we now know that there are fitness consequences for individuals, both of the individual and group social phenotypes. And despite these two levels being inherently nested, we observe different fitness consequences for individuals based on their individual or group social phenotype. So to add just a little bit more to this story, and while this isn't one of our fitness measures we're really focusing on today, I wanted to shout out the incredible work of Sophia, an undergraduate I supervised a few years back, who found that residing in more social and connected groups is associated with decreased rates of mass gain. This could be because residing in a more social group could result in more time and metabolic energy from the individual level social interactions that comprise and structure a well-connected group. And if you remember how important get fat or die is in the system, this finding bolsters the importance of group social structure for individuals. Lastly, with Sarah, an undergraduate I co-supervised in the field this past summer, we took the social cohesion hypothesis, which says more social individuals are less likely to disperse, and we extended that to the group level to ask if residing in more connected social group structures would result in less individual dispersal. We found that individuals in groups that are structured around one or few individuals is associated with more dispersal. That is, groups with high network centrality are associated with individual dispersal. That's because that central individual could be a dominant adult female or a more dominant male, kind of beating up potentially on everybody else. It's a centralized network, so more dispersal. So not only is the type of group an individual resides in influencing fitness consequences, but also these major life history decisions. So we have variation, we have replication, and we have unique individual fitness consequences of both levels of social organization. And thus, both individual social position and group social structure have met the requirements for evolution by natural selection to occur. But of course, these two levels are nested. And so maybe the results I've shown thus far and the results we've seen across taxa are detecting selection at the level measured, when actually some of that selection may have been on the other level. And thus we should quantify natural selection for both of these levels of social organization simultaneously with a multi-level selection perspective. To quantify multi-level selection, we can use a contextual analysis, which is an extension of the Land-Arnold selection analysis, and it uses partial regression to partition selection among phenotypic levels. Contextual analyses identify the levels at which selection is acting and to what extent they're acting, and they distinguish from direct from indirect selection at each level. For example, group level selection in the absence of individual level selection would indicate direct selection for the group social phenotype and indirect selection for the individual phenotype. And with these contextual analyses, we have to scale each phenotypic level in a biologically relevant way in order to interpret the selection gradients. So this is the basic structure of our contextual analysis, where the dependent variable is an individual fitness correlate, say individual summer survival or reproductive success. Now, for scaling of the measures of individual social position, we scaled the individual social phenotype within groups, within years. And for measures of group social structure, the group social phenotype, I scaled across all groups, across all years. In the measures of the individual and group social phenotype in this contextual analysis, I chose 4 pairs of analogous social network measures that tell a similar story across the 2 levels of organization, such as how isolatable an individual is within their cluster and how breakable their whole group is, how connected an individual is, how connected the group is. And these are all measures used in previous analyses I've mentioned today. And because of the long-term data set we're working with, I was able to fit all 8 of these measures into the contextual analysis at once, removing the whole pair if either of the measures had a high VIF value, as you sometimes get that with social networks, where there's some higher VIF values between some of the network measures. Now, do we need these analogous pairs for multi-level selection? No, but it sure does help simplify the interpretation if we were to find selection within these pairs, as we could perhaps find connectivity, for example, is under selection at the individual level, but not the group level, or vice versa. Otherwise, if we don't find selection within these pairs, selection across different social traits would represent that some social traits are under selection at one phenotypic level, whereas different social traits are under selection at the other phenotypic level. While I ran the contextual analyses for the 4 different age sex classes in the system, I am only going to present you today the adult female contextual analyses. Because marmot's social groups are matrilineally structured, many of the results presented here today were already adult female based. The adult females are an interesting group to focus on. So for adult females, I fit 3 separate contextual analyses, one for each of summer survival, winter hibernation survival, and reproductive success. For each model, I fit the 4 pairs of the scaled analogous social network measures. I also fit other relevant fixed effects for each model, such as predation index for summer survival or fall body mass for winter survival. Each model also had valley location, remember our higher elevation or our lower elevation sites, and social group size as additional fixed effects. And as random effects, I fit year, individual ID. And so I'm asking about multi-level selection for the individual and the group social phenotype after controlling for other biologically relevant variables. So if there is selection, it's within the context of the biology of this system. It's not in isolation. So this really is quite a simple and elegant analysis. It just requires a whole lot of data and prior knowledge about the system, both of which we have. So first, what selection is there for the individual social phenotype? On the y-axis here, we have if an adult female weaned offspring from the burrow for a given year, which is a proxy for reproductive success. And on the x-axis, we have our social measures. And so as we move along the x-axis, sociality is increasing for that particular measure. I found that degree, which is the number of social partners an individual has, was under negative selection in the context of reproductive success. In other words, this is selection for fewer social partners. And I found that closeness, which is how many social links an individual is away from all others in their group, was under positive selection in the context of reproductive success. In other words, this is selection for being more socially close to all others in your group. Taken together, these 2 results suggest the individual social phenotype is under antagonistic selection dependent on the specific social trait. This antagonistic selection could represent an evolutionary balance in this system between the negative costs of social relationships for reproductive success, while still maintaining closer social distances to others in the group to maximize potential anti-predator benefits, such as hearing alarm calls, responding to others in visual proximity, that might allow for more time and resources to be devoted to reproduction, thus boosting reproductive success. Now, what about selection for the group social phenotype? On the y-axis, now we have adult female hibernation survival for a given year. And on the x-axis, we again have the social measures. And as we move along the x-axis, sociality is increasing for that particular measure. I found that cut points, which is the number, again, the number of social ties that must be lost or broken for the group to fragment into 2 or more separate social groups. So further down the x-axis means the group is more strongly connected. And this was under positive selection for adult female hibernation survival. In other words, this is selection for more connected and less breakable groups. And I found that transitivity, which is the proportion of connected transitive loops, another measure of group connectivity, was under negative selection for adult female hibernation survival. So together, these two results suggest the group social phenotype is also under antagonistic selection dependent on the specific social trait. The cut points result suggests selection favors individuals residing in more structurally cohesive and less fragmentable groups because the breaking of groups may reduce the number of individuals engaging in social hibernation, reducing subsequent thermoregulatory benefits and increasing the risk of death in hibernation. Whereas the transitivity result suggests selection for adult females residing in less socially clustered groups. And so what this says is that the type of group social connectivity matters. You don't want to reside in the group that will break apart, but you also don't want to reside in highly clustered and transitive groups. There is a balance to the type of connectivity of the social group structure. When looking at this all together, adult females' individual social position and group social structure is indeed under multi-level selection, dependent on the specific fitness context. The individual social phenotype is under selection in the context of reproductive success, and the group social phenotype is under selection in the context of hibernation survival. This is context-dependent multi-level selection. It's important to note that most of the social traits were not under selection. And so it's these very specific contexts that the individual and group social phenotypes are under multi-level selection. And I didn't find any case for selection within the analogous pairs. And this suggests that traits at one level are also not under selection at another level in the same setting, but that this suggests that some traits are under selection at the individual phenotypic level and different traits are under selection at the group phenotypic level. And because the two social phenotypic levels are scaled, the model estimates represent the selection gradients, representing the magnitude of selection. So if we average the selection gradients, this suggests stronger selection for the group social phenotype. Interestingly, summer survival wasn't under selection at either level. And remember, selection for group living in this system may be partially attributable to predator avoidance. And thus, this might suggest that selection for group living is different than the selection for social position and structure that emerge from that social group living. Interestingly, 3 out of the 4 of these results from the multi-level selection analysis were not found when exploring either social phenotype independently. Only the group cut points measure was found previously. And this could be attributable to multiple things like different statistical approaches and different data sets. Some of those analyses I showed you before, especially the individual social phenotype and reproductive success analysis, was run over a decade ago. What I think this is actually telling us is that when we account for these nested levels of organization at the same time, partitioning selection relatively among the two levels, we get a more representative view of the nuanced selective forces acting on all levels of the social phenotype in the wild. These results not only suggest that multi-level selection occurred in the wild, but that there is antagonistic selection within each of the two social phenotypic levels. That is, there is both positive and negative selection within the individual and positive and negative selection within the group phenotype. But there is also antagonistic selection between the two social phenotypes. In other words, there is selection for more social individual phenotypes potentially being counteracted by selection for less group social phenotypes and vice versa. This complex antagonistic multi-level selection, both within and between social phenotypes in both directions, could increase the rate of an evolutionary response, or it could flatten or constrain the overall evolutionary response. Personally, I think this antagonistic selection is flattening the evolutionary response. Remember, marmots are the weird one on this list. This antagonistic multi-level selection may provide an explanation for why sociality is not as largely beneficial in this species as compared to other species on this list. This antagonistic multi-level selection flattening or restraining the overall evolutionary response may also explain why sociality is not as genetically heritable in this system as it is in others, like the rhesus macaque and humans. The rest of the species on this list are also more gregarious than the marmots. And given that at least 2 of these more gregarious species we know have more genetically heritable social behaviors, perhaps we can predict that there is more aligned and more positive multi-level selection for individual and group social phenotypes in these more gregarious species. So, the evolution of social position and structure is indeed possible with a multi-level selection perspective. We have variation, we have replication, and we have fitness consequences for both levels independently. But most importantly, we have multi-level selection for these 2 levels of social organization. And earlier, I posed you the question, how much of this individual level selection detected across species is actually group level selection? Well, the answer isn't 0. In the marmots, the strength of selection was actually stronger for the group social phenotype. So in some cases, it could be half or more of the selection at the group level. And thus, to understand the evolution of social behavior, social organization, the evolution of society, we should not ignore the group level, nor should we ignore the individual level. But we can explore these levels simultaneously. Even if we're only interested in one of the 2 levels, we can explore these levels simultaneously. Because otherwise, we may be incorrectly attributing natural selection to the level of organization, not actually under direct natural selection. Now, this multi-level selection perspective doesn't just apply to the evolution of animal societies. Multi-level selection has ranging implications. A lot of biological traits and processes are nested or hierarchical, and a lot is in the human world too. People like David Sloan Wilson are already thinking about this, from human cultures to business organizational structures and operations. An example I have for you is that we often think of human loneliness as an individual level trait, that being lonely is the result of having few social partners. But how much of individual loneliness is a product of processes at the individual level, and how much is a product of processes at the group level? A multi-level selection perspective can help us ask this question, and could even help mental health practitioners develop targeted interventions relative to the strength of the effect at each level of organization. So hopefully I've left you with many questions about multi-level selection and how it might apply to sociality and other traits in your study system. For me, 3 major questions have my attention right now. 1, what about multi-level selection in these more gregarious species, like the rhesus macaque? The genetic heritability of sociality in this species is more than double than that of the marmots. So is selection more positively aligned between levels of social organization in rhesus macaques? And what level is under stronger selection in this species? And in these more gregarious species, where we do see more positive selection for sociality on the individual level, why aren't all individuals maximally social? Why aren't all groups completely connected? Is this selection at the group level preventing this? Is there competitive exclusion? How can multi-level selection help explain this? And earlier, I mentioned that we know the individual level social traits are genetically heritable, but we don't know as much about if these group social structure, the group social phenotype is as directly genetically heritable, and how do indirect genetic effects play into both the individual and group levels. So to ask these questions, as you may have guessed, I'm now working with the population of rhesus macaques on Cayo Santiago in Puerto Rico with Dr. Lauren Brent. I'm a postdoc in her lab here at the Center for Research in Animal Behavior at the University of Exeter. This is another long-term data set nearing 15 years of detailed social behavioral data and with a pedigree and demographic information going back until the 1930s. Lauren gave an incredible Find seminar in the last series, and I strongly encourage you to go back and to watch that. And so with this incredible study system and data set, I'm delving deeper into the evolution of social behavior and social organization with this system. I'm also continuing work with the Rumble Marmot Project to continue to ask questions about the drivers of social variation and the evolution of sociality with this multi-level selection perspective. So, in summary, we know societies are complex, consisting of many levels of social organization. And exploring levels of social organization independently may detect the wrong level of selection. By using multi-level selection, we can partition natural selection among levels of social organization relative to each other. And in doing so, we see the evolution of social position and structure is indeed possible with a multi-level selection perspective, perhaps tells a more comprehensive story. We even see that group level selection may be stronger than individual level selection. And specifically in the marmots, I showed you that the timing and duration of seasons and the gut microbiome drives some variation in social behavior. I showed you that social behavior is genetically heritable. I showed you there's a mixed bag of costs and benefits to individual social position and group social structure when explored independently. And importantly, I showed you we can explore social position and group social structure together with a multi-level selection perspective. That multi-level selection might tell a more comprehensive story about the evolution of social organization, and that this multi-level selection perspective may help explain why marmots are weird. Of course, thank you very much for listening, and to Fine for hosting me. It's an honor to be here. There are too many thank yous to give, but a big thank you to the staff of the Rocky Mountain Biological Laboratory where this research happened, because they make long-term research like this possible year in and year out. And with that, I'm very eager for your questions.
All right, Connor, thank you for a really fascinating talk. So at this time, it's the opportunity to ask Connor questions. For those of you who are new, or just a refresher for everyone else, what we do is we ask you that you please put a question mark in the chat. At that point, I'll be monitoring the chat, and I'll call on you to ask your question. If you are watching from YouTube, please just post your question in the chat function online, and one of us will relay that question to the speaker. We are continuing to try and encourage participation by our junior colleagues. And so if you're a student, a postdoc, please ask your questions, and I'll attempt to prioritize as best I can while keeping an eye on the chat here. And so with that, I'm going to start with questions. And I'm going to start with the first, because it's someone new. And by the way, when you ask your question, please do tell us where you're from. Tell us just a brief little bit about you, where you're from and what your interest is. So I'm going to start with a Eyal. Did I pronounce that correctly? Rosengarten?
As much as people from America can. Hello.
So I'm sorry, you're breaking up a bit.
Bar-Ilan University in Israel. It might be because my internet is a bit unstable. I'm from Bar-Ilan University in Israel. My question was about, you talked a little bit about lifespan and the centrality of the species you're working on. And you said that the more central the individual is, the shorter the lifespan, not the shorter the lifespan, but you expect there to be a shorter lifespan in comparison to other individuals. What do you suppose causes the shortened lifespan? Because I know in rock hyraxes, at least, that centrality increases lifespan.
Yeah, that's a really good question, right? Marmots are weird. So a lot of the things we've all in this room have talked about and studied, or sometimes in the opposite direction. I don't know. I think there's some people who will hit me to say that maybe those social interactions are more costly for the marmots. It could be stress. It could be time that they're spending interacting, right? We're only observing them above ground. So if they're out, they're exposed to predators. So maybe the more connected you are, the more you're interacting, the more likely you are to get snatched up by a predator. And it could be that if you're spending all that time interacting, you're not spending as much time eating, you're not getting as fat, and thus you're not going to be able to survive an increasingly unpredictable winter. So the answer is we don't know. We don't know the exact mechanisms for a lot of these associations, but those are some potential explanations.
Thank you very much, and thank you for the lecture.
Thank you for coming.
Okay, so our next question in the chat goes to Eduardo.
Thank you, Lauren. Thank you, Connor, for a wonderful talk. It's remarkable. It's fantastic to learn about such an amazing system and all the things that you're now able to do. I may have missed very early on, as you were describing the natural history of the marmots. If I remember well, you made reference to some very mid-90s or 98% mortality. Can you remind me, when in their lives do you have that most of that mortality? When does it happen?
Yeah, so in the summers.
I was connecting that. I was just curious when you see, I mean, trying to take a lifelong perspective to fitness and survival and trying to identify the life stages where selection may be acting with 98% mortality. I was just curious at what age they are dying, because that probably should inform where we look for potential sources where natural selection is acting.
Yeah, so the 96%, so of individuals that die in summer, 96% of those individuals died from a predation incident. So the average lifespan's just above 4, I think it's like 4.06. So they're not living super long. And those are obviously for the individuals that stay. We don't really track. We know someone in the 90s kind of radio tracked them. We know they go and they disperse way far out of where we study, but I don't know what the average lifespan is for those that disperse.
And a follow-up, an age-specific mortality, or like age trajectories of mortality, because with such a short lifespan, knowing when they are dying, I'm just curious, when are they dying?
Yeah, so I don't remember what the exact average litter size is per mom per year, but say it's 3 or 4. I'd say most, maybe the mom is lucky if one of those survive till yearling the following year. So most are dying in their first year. And so the networks and the results I just shared don't include pups. They emerge halfway through the year. The dispersal just kind of happened. They're tiny. The grass is way tall. So the results we have for this isn't including the pups. So most die in that first calendar year. They don't survive to their first hibernation, or they don't survive their first hibernation. And so that average lifespan is incorporating that. So individuals who do survive past that first year are probably actually living maybe 6 or 7 years.
Thank you.
Okay, our next question is Raghavendra.
Hello, can you hear me?
Yes, thank you.
I'm Raghavendra Gadakar from the Indian Institute of Science. A train is about to go by. I will pause for a minute if it does make a noise. Thank you very much. I really enjoyed this talk. I think I finally understood the multi-level perspective, which I've sort of been trying to wrap my mind around. But the proof of my having understood is based on the question I'm asking you. So if I've understood you correctly, both the concept of multi-level selection and your results, it would mean that the same individual with the same phenotypic traits would have different fitness depending on whether it will be in group A or in group B, if the groups A and B have different group level features. Is that a reasonable conclusion?
Yes, I think that's reasonable. Yeah.
Okay. Now, what do you think are the prospects of proving this experimentally in your system or any other system if you have a large enough group and hope that moving one individual doesn't change the group level features of that individual, but just exposes the individual to a different group? This occurred to me when in your last slide, when you talked about humans. It seems to me that obviously one of the therapies is you change your group composition. You move somebody from one classroom to the other, one group to the other. So what are the prospects of doing this in any animal system and proving that when you get a result such as yours, we can actually demonstrate that this is true?
Yeah, so I think if someone is here who knows the paper I'm about to talk about, please jump in. I think it was in iScience within the last year. It was maybe vervet monkeys that when they watched as they dispersed from their natal group to another group, those individuals kind of adapted their sociality, is the term they used, to the group that they joined. So if they were, to really oversimplify this, introverts in the group they're coming from, and they went to a really social group, they became more social in that group. So is that a product? How, I mean, obviously there has to be some social plasticity there, but how much of that is actually right? If they put it as a cultural perspective. So
the flip side of that is that, if the flip side of that is that if they don't change their social behavior by moving to another group, would they have lower fitness? That's the, I mean, if the individual maintains the same feature, but in this environment, the environment now being the group social environment, I have less fitness, and in this environment, I have higher fitness. Do you think this can be tested experimentally in any system?
I mean, that would be really, really neat. I'm most certainly not an experimental biologist, so my system would be flawed in setting this up.
Yes, but some other system, maybe, I don't know.
Some other system potentially. I think that'd be difficult because what is the individual phenotype and how plastic is it? Is it, if we move a really social individual to a less social group, is that social mismatch? Are they able to adapt or be plastic in their new group? I think it'd be really neat. I envision some challenges.
Okay, thank you.
Okay, so our next question is Filippo.
Hi there, Filippo Aureli from Universidad Veracruzana in Mexico. And Connor, I really enjoyed your talk. It was very well pitched. I think you repeated several of the concepts along to make sure that people like me will be able to follow. So I really, really appreciate it. Thank you. I have a couple of questions. One is a quick comment on the last question of Raga, is that I feel that if we take this very nice link between individual level, group level, all the way, I feel like that the experiment will not be able to do it simply because by moving one individual in a new group will change the group level structure as well. So I think maybe that would be possible only with kind of simulation, like in a way controlling for factors and so on. So, but in real life, let's say it'd be possible, as Connor was mentioning, that experiment, that an individual will adjust. But in principle, by moving anything, will change the two levels because of the connectivity between the two. My question, I don't know if you agree, Connor, on this.
I do.
Okay, yeah. My first question is rather short. And then I would like to ask the last question, but I see if there are more questions, I can wait for the next turn. It's a more technicality. You mentioned that you were focusing today on presenting data on the affiliative networks and not the agonistic networks. But if I understood, your affiliative network is basically based on one measure. It's not they are not all the various behavioral affiliative behavior together, but it's just kind of proximity or something like that, if I understood correctly. But I may have missed at that point.
No, that was my fault. I did not explain that well. It is all the affiliative behaviors, whether it's play, grooming, foraging together. It's all used together, classified as just affiliative.
Can you explain me how I could check probably on the paper, but how you cluster them together? How would you do it? You just sum them all because they are measured, they are collected all in the same way. Okay.
Yep. So it's just a weighted network. So any affiliative interaction, we just ticked off one. So if they engaged in grooming 5 times and a play behavior 5 times, that's 10 affiliative interactions.
Perfect. Okay. Thank you. Okay, that's good. Now, instead, my question, the real question is on the multi-level that was fascinating in the way that you explained it and so on. If I understand correctly, your results. So in principle, it could be you could have an effect of one particular measure of a dependent variable, let's say, affected in one way from any individual level characteristic and in a different level of the group level. But you don't find that, if I understood. You find basically the contrasting were within the same level. Like, so they say you found that the contrasting selection on the same dependent variable within the same level. Okay.
Yeah, so like reproductive success, only individual social behavior, hibernation, only group.
Exactly. So would you then I mean, I think it was very nice that you have done it all, but let's say to get the same result you got, you could you have done it within the same level without having the multi-level approach? Or you think it by controlling for the other level, you found this within level? Because I believe you could have done it, you know, listening to you is okay, you could do it only, instead of looking at one single variable affecting the dependent variable, you could have seen several at the same level, at the individual level, let's say, and you would have found possibly the same results. But maybe not, because you want to portion out the group level, or the other way around. So just, I don't know if I'm clear on my question, but yeah.
Yeah, so that was what the middle of the talk was, and it's just me poorly describing it. So we've done all of this individual level traits. So I just shared very quickly on one slide all the individual level stuff. And so that was fitting multiple affiliative individual level social network measures in the same model and just looking at the individual level. And those were the consequences of that. And then I did the group level, and then I put them together. So you absolutely can. But the results when you explore it independently, and the results when we explore them in the same model, that multi-level selection model, the results were different.
Yeah.
That could be because the data sets are different sizes. Some of those analyses were done a decade ago. Could be because it's a different statistical approach and we're chucking more things into the model. Or it could be because some of that stuff they found in the last decade on the individual level is actually group level processes, or vice versa. I'm more posing that to this community than providing you with a very specific, it is at this level or is not at this level. These are associations.
Yeah. No, thank you. I didn't understand the results of the same level that were with multiple independent variables. So that's why I thought it was. But well, no, fantastic. Thank you for the answer.
Okay, so our next question goes to Sari.
Hi there, I'm Sari van Balle. I'm at the University of Texas in Austin, and I study the social system of black howler monkeys in Mexico. I really liked your talk, just like what Filippo just said, well organized, and a person like me with little background in this multi-level selection theory could understand. It's still, I have a hard time wrapping my mind around the group level selection idea, given that it's selection on individuals, right? I mean, and then you said that it's such a great turnover. If the lifespan is only like something like 4 years old, there must be a great turnover from year to year. Those marmots emerge, and there's not all of the individuals that were there last year are there, and so they have to establish, it seems, new relationships, new bonds, obviously based on what they had the season before and that. But still, how can that, let's say that we had 4 individuals in season A, and now the season B, the next one, we only have 2 of these, but we have 2 new ones. They immigrated, or like they are pups that grew up. How is it? It seems to me that they have to establish a new group with its closeness and the network and all the measurements that you can measure on the group level. How that is that then, can that be selected for? How can that be inherited from year to year if these individuals change so quickly? So I can't wrap my head around selection on the level of the group. I understand that there is selection on the level of the individual, and they have interactions, and from there emerges group level characteristics. But how that leads then to group level selection?
Yeah, that's an excellent question, because they do go down, right? These groups don't exist for the whole year. They go down for hibernation for 6 or more months every year. So the groups do restructure every year when they reemerge. Group membership stays somewhat similar. You're right in that some yearlings are recruited. The more socially connected a yearling is, the more likely they are to be recruited to their natal colony. Everybody else kind of goes away, disperses. I'm forgetting exactly what paper it is from the 60s or the 70s that was saying that when group turnover, so like when these groups do restructure, if groups restructure more often than like the average individual lifespan, group selection can actually be stronger than individual level selection. So our groups are turning over yearly, as where our individuals are living until about 4. And so that what we found there does align with that. I'm very sorry, I'm blanking on the paper. I could point you to there. While the average lifespan is 4, the groups are formed, they're matrilineal. And so the adult females can live up to, I think, 13, maybe 16 was our oldest one. And so really the core individuals, sometimes there's multiple within a group, that are structuring these groups, that are kind of the central folks, those are maintaining from year in and year out. So while the groups are restructuring, they are starting to restructure kind of in the same pattern, but you're absolutely right. Group sizes are changing, environmental conditions are different. And so if we think having a group that doesn't break apart is good for hibernation survival, every year the group should restructure in a way, if there is positive selection for that, in a way that the groups don't break apart. And those groups that do break apart, that's why we have less hibernation survival for them. And so, yeah, group membership changes, but those structures can come back year in and year out because the individuals sometimes are the same.
All right. Lovely talk.
No, thank you.
Congratulations.
Okay, so our next question goes to Tony.
Hi, I'm Tony DeFury from the University of Texas at Austin. I study South American primate social behavior, social evolution. Connor, thank you. I want to echo Sari's last comment there. It was a great talk. I really enjoyed it. You were super clear in how you talked. So I very much appreciate that. And I think Sari articulated kind of the question that I had, probably in a more specific and less fuzzy way that I'm about to articulate it. But two things I guess I wanted to ask you about. First is, these, both at the individual level and at the group level, these social phenotypes that either an individual or a group has are composites of lots of different things, right? Lots of different behaviors go into them. And as you nicely pointed out, the phenotype at one level can influence the phenotype at another level. So when we think about selection asking, I guess I'm posing this question to you. What's the individual phenotype then that is either being selected for or not selected for? Is it a propensity for prosociality? Is it a propensity to be tolerant of other individuals near there? So what's kind of the proximate level phenotype that is, presumably tied to genes in some way that's giving you that heritability? So first, maybe speculation about that. And then the second question is, that I liked your, I thought very clear explication of the model that you used in the paper that you and Dan and Philson, I think, it's you and Dan and someone else have in review right now. But I'm just wondering, to what extent is it necessary to posit that there's a genetic component at the group level as opposed to an environmental component that's at the individual level, where the environment is the social environment, right? So an individual's genotype and phenotype expressed in a particular environment, where the environment is your social group. Why do we need to go? I guess this is a philosophical question, maybe an empirical one too, or a practical one. Why do we have to go to saying there's a genetic component that we need to account for at the group level, as opposed to that kind of, heritability is a product of this phenotypic variance and genetic variance and their intersection, right? Or environmental variance and genetic variance and their intersection. So those are kind of two questions.
Yeah, no, excellent questions. So I like to joke that the marmots, the ideal marmot is going to go to a party, but is going to stand in the corner and not really interact with anybody at that party. They want to live in the social group to reap maybe the ability to not have to disperse, or to gain those anti-predator benefits. You trust, who's alarm calling, you trust those alarm calls. So they want to be in the group and they want to be somewhat connected in the group, but they don't want to be the most connected in the group. So there's this balance. And so that's where we saw negative selection for degree. So you don't want to be connected to a lot of people, but you want to be socially close. So maybe I want to be connected to the most social individual who's then really close to everybody else. So I have that next level away, my immediate indirect partners are quite large, but my direct partners are quite low. So I think it's a really specific type of individual level connectivity that the individuals want. And then your second question is kind of asking about, I think, the relationship between individual measures and group measures, right? The group feeding back to influence the individual can be perceived, right? We call these things the social environment. It's a term we all use. So you can absolutely explore how, right, in a highly dense group, a group where almost everybody is interacting with everybody, that's going to drag up the value of individual connectivity. But the distribution of individual level behaviors can be different. To get a density value of 5, where half of all the possible connections are observed, you could have just a few super social individuals who are connected to everybody, and then, you know, a kind of almost a beta distribution where then some individuals aren't really connected, or you could have a more normal distribution. So the distribution of individual traits can be influenced by the group. So that's another point I didn't really make in the talk that needs more research, is decomposing specifically what distributions of individual behaviors result in what groups, and what group structures and how exactly they feedback influence the individuals. So the answer to your question is absolutely yes. I think we could explore the group social phenotype as an environmental driver of the individual phenotype to really, and networks can help us do that, but networks most certainly aren't perfect.
Okay, so I think I'm next in the queue. Lauren Hayes, as I told you earlier, I'm the host. I'm at the University of Tennessee, Chattanooga, and I'm interested in fitness consequences of group living and also social evolution of mammals. I think my question is connected to the previous two. And I apologize, it might be naive, but I'm trying to connect the two. And is there some level of like homophilic associations that occur between these animals? So do you have a preference for a certain phenotype like AGD? And could that potentially influence the cost and benefits of group living?
Yeah, so one of the measures, I didn't talk about all the global group level network measures I used. There's 6 of them. One of them is degree assortativity. And so in that, we would expect higher values of this degree assortativity value would be more socially similar profiles. So extroverts hanging out with extroverts, introverts hanging out with introverts, whatever it is. That didn't stand out any of the models. So that social similarity, at least at the global level, isn't mattering. Now there is, right, they have preferential partners. Those females, year in and year out, when they emerge from hibernation, reaffiliate with the same individuals in their group. So that on the individual level does matter. We're just not seeing that kind of assortativity, homophily scale to the group level.
Okay. Thank you. I think I have to think about my next question because I'm thinking about relative costs and benefits of different forms or structures. You know, may not be in an overall model, it may look the same, but if you look at it more specifically, are the slopes different depending on that? And I think your answer would be no. Or
yeah, I mean, I think what the multi-level selection was saying, because those are both the cut points in the transitivity measures that popped out. You can broadly classify as both measures of global social structure connectivity. So the fact that one is positive and one is negative is contrasting, right? It's interesting and why. So it's really, there must be some specific perfect group structure that exists for a given year and a given composition of individuals, and when the snow melted and how much food's around and how many hawks are flying ahead. There must be some structure that's perfect. And we know, at least from this, or we can think at least from this, that you want a group that isn't going to break, but you don't want a group with a lot of connected triads. So there's a specific connectivity that is good and a specific connectivity that's bad. Are marmots aware of their group social structure? We don't really know. Like a theory of mind, how much are they aware of? How much am I aware of my network? I don't really know who my friends interact with. So it is quite a difficult scaling to the group. It brings in so many more layers of complication.
Sure. Well, thank you. Okay, so our next question is Mateus.
Okay, you could listen.
Yes.
Okay, well, first of all, I could say that your presentation is very interesting. I will have the opportunity to read the papers, and it's interesting the topic. But I'm from Colombia, to the Universidad del Rosario, and this is, well, my question is, which behaviors are most beneficial for reproduction in segment groups?
Yeah, so which behaviors at the group level are most beneficial for reproduction? Nothing stood out. I'm trying to think. We know that, so in the multi-level selection, only individual level sociality had a relationship, is under selection for in the context of reproductive success. When we explored the group independently before we put it all together with multi-level selection, groups that are more easily breakable, right? We think that's bad for hibernation, but that was good for reproductive success. And that's maybe because a group that is breakable is potentially spending time in kind of two kind of separate groups with some connections in between. So these groups are kind of breakable. And maybe that means the females who are the reproductive success we explored in aren't having to spend as much time interacting or aren't subjected to that many interactions. They can spend more time investing in their offspring. They only have so much time in a day. So in the multi-level selection model, nothing at the group level popped out for reproductive success. But when we broke it down independently, cut points did pop out just in the opposite direction from the hibernation survival, which again is conflicting because reproductive success, you can't reproduce if you don't survive winter. And so it's complicated, sadly. I wish it was a clear answer.
Great, thank you so much.
Okay, our next question is from Karsten.
Yes, hello, I'm Carsten Schradin from the CNRS in Strasbourg, France, studying mammalian social evolution, currently at the Succulent Research Station in South Africa. Thanks for a very stimulating talk that made many of us think and maybe reconsider multi-level selection. And I will have to watch the talk again. Probably my question is not very correct, but like Sari, I still have problems understanding how the selection on the group level should be that important. Stimulating is we had on Saturday here at the Research Station Journal Club where we discussed Hamilton's great paper from 1964, and now I better understand also the title, The Geometry of the Selfish Herd. And I was just thinking your main, maybe there were other measures, your main measures of the group level was the connectivity of groups. And Hamilton in his paper, he develops his theory by looking at connectivity between neighbors in his first part of the paper, totally independently of genetic relatedness, and later put this in. So I was wondering if this connectivity, he calls it how everybody influences each neighbor and every neighbor influences each other, if that is not merely the same like how connected groups are, and that if one would want to, and mathematically it's the same, one could explain it also on group level selection, but on inclusive fitness and kin selection.
Yeah, I mean, in my head, I envision a day where I'll have a beautiful Venn diagram that is just, we need to decompose the individual and how patterns of individual connectivity, what type of groups those specifically structure. Because if you are selecting for increased individual degree, you're of course going to get more dense structured groups. So where that Venn diagram is, so if you have this selection at the group level, how independent is that of the individual level, right? It's not the fact that there's different directions of relationships implies that it's not a perfect one-to-one, but where and how much that Venn diagram is overlapped. Because you're right, is the selection at the group level has to go back down to influence patterns of individual connectivity. So maybe while the estimates for groups could be inflated, and actually we need to somehow scale those estimates in a way that's more reflective of that it's actually potentially more individual selection, but some of that's feeding through from the group level. So the answer to your question is, I think yes. But how exactly and with a network approach, I don't know if we know exactly.
I mean, after the price equation, higher level selection and inclusive fitness is similar. And the question is always and has been, how can we explain it better? And I think in your talk, you could show very well with your graphs and your analysis how you can use this higher level selection to explain it better than it would pop out of my head now, how I could explain it by inclusive fitness. But at least if I think like it down for me, it's easier to understand why this is possible.
Okay, thanks for that. And so our next question is Anna-Marie.
Yeah, hi. Thanks for a great talk. My name is Anna-Marie van der Marel, and I'm a postdoc at the Universidad Católica in Chile. My question is kind of like how you defined social groups and how you created your networks, because I can imagine that the affiliative interactions that you used are not only between individuals from the same social group, it can also happen between individuals like within the same population. So yeah, like how did you kind of account for those other interactions?
Yeah, great question. So where we study them in Colorado is at the base of a valley. They live in colony areas. And so those colony areas that we study are spread across about 5 kilometers at the base of this valley. Marmots are great to study. I used to study birds, and then I studied marmots. Marmots have a zip code. They live in one place. They don't really move, which is great. Those same individuals live within a colony, and they don't really interact within the colonies. Two of the colonies are semi-close, and sometimes a male will run between them. And so in those years, we kind of just treat that as one colony. But the colony doesn't really matter. So they're not really interacting between colonies. They're not moving between these colonies. But within a colony, you're right, there are and can be multiple groups. So there could be groups that live higher up on the slope and some that live further down the slope or left and right. And so we use a path analysis, I'm trying to think of the exact name. It's called map equation. It's like a community detection algorithm. It's not a path analysis. It's a community detection algorithm that tells us based on shared space use, who's in a group. And so we give it all the interactions in that colony. We actually just do it, all of the up valley sites and all of the down valley sites, and it spits out essentially what you would expect in a colony. And so we use that to define the social groups. So within a colony area, even if there were a few interactions between what it spits out as 2 separate groups, even if there were a few interactions between them, they weren't really sharing space that much. And that community detection algorithms, like those were one-off interactions. They're not really its core social group. So how we define it is, who are you spending the most time with? Who are you sharing the most space with?
So in the end, it is indeed like social groups that have been gone into the social networks.
Yeah,
exactly.
What is a social group is my favorite question. I still don't know exactly. I think this is a not wrong approach. I don't know if it's the perfect approach.
No, and I have actually a second question, if that's okay to ask. So it's kind of interesting that the summer survival is not really under like the multi-level selection, or at least you didn't find anything. And you mentioned that anti-predator behavior is super important in these for summer survival. Then also within thinking about also like how the colonies are how the social groups are within those colonies, are individuals that alarm call within the colony, can do all the other individuals within the colony hear it, or is it just only really the social groups?
Yeah, so I'm thinking of, yes, they're really loud. So everybody in that social group can hear it. I can hear it from a couple hundred meters away. They're really loud. They're really high pitched. So thinking of a slope in the Rockies, if we call this one colony area, and then with our community detection algorithm, it's going to spit out, maybe there's a social group at the top and a different social group at the bottom, infrequent interactions between them. So we can be confident that they're calling them separate social groups. If you alarm call up there, I can definitely hear it down there. And so the alarm calling can travel. You can hear that across separate social groups.
Well, would it then be for summer survival, not be better to look at the colony level versus group level?
Yeah, so could we build a network on the colony level? I think that's the analysis I'm going to do tonight now. So yeah, I think that's a great question. I think if thinking about summer survival and if the benefits are mostly, we expect to be mostly predator related and alarm calls kind of their primary mechanism for that. I guess the social group could be, and maybe should be, the maximal distance that you can hear an alarm call.
And then it will be difficult to find what's the maximal distance to hear.
Yeah, and then the social group will be much more sparse because you are going to have you're going to have a big clusters within the group, and then by not using the community detection algorithm, you incorporate those sparse interactions between. So I would expect kind of a big cluster here with 1 or 2 interactions between, holding them together. But if you're doing that for all the colonies, there are some colonies that only have the one group. But by incorporating that and basically adding to the distribution less connected groups, it could spit out, because we know there's benefits for the individual. More connected individuals have better summer survival. But yeah, the group, maybe we're thinking about groups the wrong way.
Interesting. Thanks.
Thank you.
Okay, our next question, we have another one from Filippo.
All right. Well, I follow up a bit on this last point, although I didn't agree, I didn't arrange to do that with Anne-Marie. But and linked to my first question, there was what was the measure that you had, and you say that you have basically combined the various behaviors to get this composite measure. So I wonder, maybe related to this distinction between who is in a group, who is in a colony, and so on, let's say if you would put agonistic behavior, you may actually end up to have the picture much clearer, for instance. But the question mainly is if you made you have a fantastic database in a number of years. You need to look at the various levels of selection using different measures. But one thing you may have to compromise was at the fine social level, that because you actually ended up to combine them. Maybe you didn't have enough to do it separate. Because I was thinking about maybe some nuance could be understood by looking at multi-layer network instead of just the aggregate network that you basically ended up to use. And since now more and more we are going to see that actually the multi-level network is playing something different than the aggregated and so on. So, and I'm kind of working in that direction. That's why my question is, I don't know if you have done anything, but otherwise, what you would think would be different, and potentially looking at different system. Now you're looking at the rhesus macaques and so on, that maybe is easier to do than that approach.
Yeah, you're absolutely right. Is by aggregating all of their affiliative behaviors, and they have quite a few, into one, potentially we're flattening out a potential relationship by saying play and grooming and foraging together and sitting together, they're all the same thing. But maybe being really connected in your play network, being really, foraging, being able to forage next to everybody is actually beneficial, and all this other stuff doesn't matter, and thus we're not detecting an effect. Could we pull that off in the marmots? So the interactions are relatively sparse. Now, studying macaques, I'm overwhelmed by how social primates are in relation to how social the marmots are. You can sit out there for 4 hours in the morning trying to watch marmots and only observe 2 or 3 dozen interactions, and with the primates, I'm immediately overwhelmed in 5 minutes. So do we have the power in order to decouple the types of affiliative behavior? I think that's a great question. I think maybe now, with almost 20 years of that data, maybe the models could fit. But you're right, it's absolutely, there could be a specific type of affiliative behavior. The agonistic behaviors are really interesting too. Those groups are much more centralized around kind of the adult females, kind of the powerhouse, or the adult male is kind of central, trying to beat up on everybody, especially trying to beat up on the yearlings. Even if it's their kin. I want you gone. I want the direct fitness, not the indirect fitness. So those are really sparse too. And the problem is we watch them in the spring when it's just snow, and they're just brown blobs, so we can see them really well. But then the snow melts and the grass starts to grow really quickly. And marmot, they get up to 7 kilos before hibernation, but the grass gets quite tall. So as the year goes on, they become less social, but we're also observing less. So it can be quite difficult to decompose that stuff.
Okay, so we got time for, I think, 2 more questions, and then I think we'll end it there. But great discussion, by the way. Eduardo, you had another question you wanted to ask?
Second, because I wrote it down here. Now I'm getting all confused. Yes. So, and I shared with some of the comments before that you were so clear. I think it was Sari who was saying this before, that for some of us, I'm listening to you and I'm going back to my early days of doing PubGen. And I mean, I don't know what, I mean, it's not, I mean, thinking about coefficients of selection, that's not something I do regularly. So it was very, very clear, very easy to follow you until you got into the details, and I got a little bit lost there. And where I get a little bit lost is in how it's also making me think a lot about when I try to read papers that come from the health sciences and medicine, and they're doing the kinds of things where maybe instead of looking at coefficients of selections, they're looking at patterns or percentages of mortality with different traits, etc. If I followed you well, and I was taking some notes, some of the R squares you report are remarkably small. That's why I was thinking about the health sciences. These people tell us there's a 60% decrease in mortality of this. What they don't tell you is that mortality to begin with is 0.000% of whatever. So for example, I just want you to help me. I mean, it's fantastic because suddenly I'm learning about a new topic, but the gut microbiome, and I continue to have lots of questions. What are we learning from it? The R square, I mean, this is describing less than 1% of the variation. If we add on that mortalities happen, 98% of them are dying soon, etc., etc. And then, so the gut microbiome is an order of magnitude smaller than the R square for the reproductive success of females and connectivity. So is it possible that we're just focusing that is all valid, but what we need to make clear is that this is a tiny, tiny percentage of what really matters to them, which is some of the questions that we've had following up. Is Sari's point of saying, well, the groups are changing. So yes, for the tiny little bit that sociality and microbiome is important in the marmots, then we find this. But in trying to understand the marmot as a living organism, shouldn't we be checking on why? I mean, the selection is more important when they are dying in the first summer because they're not getting fat. We should probably be looking at the adipose genes, not the connectivity genes, if we're trying to develop an argument about the evolution of everything else. That's why I'm getting confused. I don't know if I'm getting confused or if I understand it. And it is just that if we're looking at sociality in the system, it's going to be a tiny, tiny percentage of the whole explanation.
No, I think you understand it very well. This obviously fine. It's all about social behavior. This is all the social behavior stuff. But of the portion of the pie of, being a marmot, sociality is a small sliver. And so then breaking that small sliver down even more, you're absolutely right. And so there is a paper that got published in Journal of Mammalogy out of this system recently. And the R squares for the association between the microbiome and mass gain rate were much larger. I don't remember exactly what they were. And that makes more sense, right? If my microbiome is going to influence how much I can digest and store fat. And so that is a much more direct relationship. The relationship between the microbiome and sociality is probably acting through multiple steps. And so maybe it's not a direct association between the microbiome and sociality, but it's an association between the microbiome and mass gain and like these kind of multiple steps of the relationship. So you're absolutely right. I think you do understand it, that the portion of the pie of marmot fitness and survival and persistence for sociality, small. And then when we break it down more, it's even, that's why the R squares are small. I think they're important relationships and associations to have. It'd be great if we could go and do some experiments. We don't do experimental analyses. We don't do experiments with this system. But you're right. The overall piece of the pie of sociality and the microbiome is tiny, but nonetheless, it's still there.
Right, but then shouldn't that be brought to bear on our attempt at understanding the evolution of sociality? Because it seems that really, I mean, those who are going to be social are those who are succeeding getting fat, period. I mean, most of the selection ought to be acting on something else. Filippo wants to say something. John, jump in. I mean, we're few. This is the fun.
I just say not to be eaten is the sociality advantage. So it's getting fat and not to be eaten. As Connor mentioned many times, that it's basically look like that the best is to be in the group, not too much connected. Right. So they are not really that keen to be social. Is to be in the aggregation. Probably the idea that Anne-Marie mentioned about the colony could be working because they want to be with others around to be protected by the predation.
And again, because this is I mean, I like the last 10 minutes because we're just a few and we can chit chat. And so I'll go somewhere else. But so you're saying that there has to be animals moving between communities. There has to few, but otherwise I cannot imagine these are closed systems or they would be all inbred. So how does that happen? What do we know about some gene flow, whatever that is, and how frequent and how slow the gene flow between communities? When does it occur?
Yeah, so just like individuals disperse out of what we study, individuals disperse in. So we don't know where they're coming from. They don't have ear tags. They don't have paintings on their back. We don't know where or how far they came from. Then those, especially males, will come in, they fight with the natal male. Someone wins, someone gets to stay. They are pretty, like we have seen, right? A male will try to reproduce with their granddaughter. So they're weird. And they are, I think, quite robust to inbreeding, but obviously they're not perfect. So we do have individuals coming in, I think, to kind of bolster some of that. Rarely, maybe once a year. I think right now we have like 300 marmots in the population we study. Maybe once a year is it dispersal within the area we study. But if like 100 individuals are dispersing, most of them we never see again.
And remind me, how much are they weighing? I mean, an
adult? Right before hibernation, they get to like 7 kilos. I think when they emerge, the adults are probably 2,500 kilos, 3,000 kilos.
No, cannot be
quite a lot.
No, cannot be 3,000 kilos. 2.5.
3,000 grams. Sorry. So 3 kilos. I might say 300 kilos. Imagine
that would be a marmot I'd love to see.
A new Hollywood movie. Sorry.
Attack of the Marmot.
Thank you. They're going to win.
I'm going to allow one more last question and before I stop the live stream. And I apologize again. I didn't catch how to pronounce your name because you cut out when I called your name. It's
Eyal.
Eyal. Okay. Pleasure to meet you. Eyal.
Thanks. Nice to meet you. My question is regarding you talked about the dispersal of younglings from the natal group, and my question is, did you see any bachelors orbiting the group you were studying, and if so, how do they affect your study, the calculations you performed?
Yeah, great question. So we get that in dispersal. When dispersal happens, right, individuals come in and they might hang around for like a day or two, feel like that male's really big, I'm not going to be able to move that guy up. And then they keep moving down and they keep looking for a new place to disperse into. And so with how we define the groups, with these community detection algorithms, because of the whole 5-month season, or even if we're just looking at 3 months, they come in, they're there for a day or two, maybe a week. They're not sharing space enough. They're not interacting enough to be detected and be called part of that group. So we could do, and I think it'd be really neat. I don't know, again, it's a question of like how sparse would the data be, but looking at those interactions, who do they come in? Who do they interact with? The challenge with that is going to be, they're really hard to trap sometimes. And if they're only there for a week or two, we're not going to feel confident enough to say it's the same individual lurking around. And because it's so rarely a marked individual going to a different group and playing Bachelor and then moving on, that'd be really neat. And I think that's a whole area we can explore, but I'm sorry, I have no answer for you.
That's quite all right. Thank you very much.
All right, so I'm going to quickly stop the live stream before I do. Want to publicly thank you, Connor, for a really engaging talk, and you can tell by the discussion, engaging discussion as well. This is what we love about the Find, so thank you.
Yeah, thanks for having me.
And next week we have Elise Houchard from France, I believe, talking